Hello friends, welcome to my YouTube channel Coach Up Academy, and today we are going to learn about mutation. So what is mutation? So simply we can say देखो एक organism है, every organism has their own genetic material. उस genetic material में कोई भी changes हो जाना, sudden changes has takes place और वही changes inheritable भी हों, तो इसी को हमने mutation कहा. Simply अगर हम लोग define करें तो what we are defining that a sudden stable and inheritable change in the DNA sequence of an organism is called mutation. What it is called, हम लोग फिर से define कर लेंगे mutation क्या है. A sudden, stable and inheritable change in the DNA sequence of an organism is called mutation. अब ये जो भी organism है इसके DNA sequence में changes आए हैं जो कि inheritable हैं, तो whatever the organism, whomsoever the organism is there, जिसमें सारे चेंजेस आए हैं उस ऑर्गेनिजम्स को हम लोग डिफाइन करते हैं एज अ म्यूटेंट. व्हाट वी कॉल देम एज अ म्यूटेंट. इफ समवन आस्क मी व्हाट इज म्यूटेंट, तो वी जस्ट डिफाइन दैट द ऑर्गेनिज्म हु अंडरगोज द म्यूटेशन. ऐसा ऑर्गेनिज्म जिसमें म्यूटेशन हुआ है, वी कॉल दैट पर्टिकुलर ऑर्गेनिज्म एज अ म्यूटेंट. नाउ किसी वजह से भी हुआ होगा, तो द फैक्टर्स, कोई भी ऐसा रीजन बन सकता है. द फैक्टर्स विच आर रिस्पॉन्सिबल फॉर कॉजिंग द म्यूटेशन, वी कॉल देम एज अ म्यूटाजेंस. वी कॉल देम एज अ म्यूटाजेंस. तो वी जस्ट डिफाइन व्हाट आर म्यूटाजेंस. द एक्सटर्नल फैक्टर्स विच कॉजेज द म्यूटेशन. ऐसे एक्सटर्नल फैक्टर्स जिनकी वजह से म्यूटेशन हो रहा है, वी कॉल ऑल दोज फैक्टर्स एज अ म्यूटाजेंस. अब दीज म्यूटाजेंस कैन बी रेडिएशंस भी हो सकती हैं, there can be some chemicals as well, and temperature also. So the radiation, chemical and temperature, these all are the examples of mutagens. Okay, now one more thing, that for the very first time the word mutation or the term mutation was used by Hugo-de-vries in the year 1903. So minutely we just recall the things which we studied till now. हम लोगों ने define किया what is mutation. तो a sudden, stable and inheritable change in DNA sequence of an organism is called mutation. What is mutant? The organism which undergoes mutation, that is called mutant. What are mutagens? To the external factors which causes the mutation. It can be radiations, it can be chemical, or it can be temperature. And the word mutation for the very first time, or the term, was given by the Hugo-de-vries in the year 1903. Now we come on to the types of mutation. What are these types? अब हम लोगों ने कहा कि चेंज होना है, सडन चेंज होना है. अब सडन चेंज हमें पता है, जीनोम comprises of the chromosomes, and on the each and every chromosome there are the number of genes are present over there. अब जीन्स में भी genes are also there, chromosomes are also there. So some changes takes place in the structure of the chromosome, whereas some changes takes place in the genes, the segments which are present over the chromosome. तब जो changes हमने कहा, तो that means ultimately two conditions are there for us. कुछ changes हैं जो genes में होने हैं, कुछ changes हैं जो chromosomes में होने हैं. तो on that basis we classified the types of the mutation, that the one is gene mutation and the another one is chromosomal mutation. तो what the gene mutation is, by the name itself हमें समझ में आ रहा है, the changes takes place in the structure of gene. जीन के स्ट्रक्चर में whatever the changes has been taken place, जो भी changes हो रहे हैं किसी भी mutagens की वजह से, these all are called the gene mutation. Now the chromosomal mutation, whatever the changes, or we can say the changes in the structure and number of chromosomes. यहाँ पे number भी vary करेगा. So here we define the what is the chromosomal mutation. The change in the structure and number of chromosome is called chromosomal mutation. So again we just define both the definition. We just recall what is gene mutation. The change in the structure of gene is called gene mutation, and here the change in the structure and number of chromosome is called the chromosomal mutation. When we are talking about for the gene mutation, the gene mutation is further classified. On what basis? We know that the genes हर एक सेगमेंट है, there is a specific segment which is responsible for expression of any of the character. तो ये हर एक character जो है तो DNA में हमें पता है that DNA comprises of the polynucleotides. DNA is the polynucleotide chain. तो ये जो न्यूक्लियोटाइडिक सीक्वेंस हैं, ये जो न्यूक्लियोटाइड सीक्वेंस हैं, तो द वन कंडीशन की जीन के स्ट्रक्चर में चेंजेस आ रहे हैं, दैट इज द म्यूटेशन फॉर श्योर. तो वन कंडीशन दैट कि इस न्यूक्लियोटाइड के किसी सिंगल न्यूक्लियोटाइड के स्ट्रक्चर में चेंज हो जाना, दैट सिंगल न्यूक्लियोटाइड और द सिंगल नाइट्रोजनस बेस पेयर के में चेंजेस आ जाना. सेकंड थिंग, देयर इज अ एडिशन और डिलीशन ऑफ द नाइट्रोजनस बेस पेयर्स. टू कंडीशंस आते हैं. फर्स्ट, change in the single nitrogenous base pair. Second, addition or deletion of the nitrogenous base pair. On that basis, we classified or we gene mutation into the two categories. The one is point mutation. The second one is frame shift mutation. The point mutation, by the name itself, हमको समझ में आ रहा है point by that means single point. So the changes which take place in a single nitrogenous base are called the point mutation. Right? There is a changes in the single nitrogenous base. सिंगल नाइट्रोजनस बेस में जो चेंजेस आएंगे दैट इज कॉल्ड एज द पॉइंट म्यूटेशन. एंड फॉर दिस द मोस्ट कॉमन एग्जांपल इज सिकल सेल anemia. This is the disease which has been occurred only because of the change in the single nitrogenous base pair. We come on to the next. तो frame shift mutation. What is frame shift? तो यहाँ पे हम लोगों ने डिस्कस किया that the addition or deletion of one or more nitrogenous base pair. Here we are talking about the change in the structure. उसकी position change हो गई है. But here we are talking about the addition or deletion of one or more nitrogenous base pair called the frame shift mutation. For example, यहाँ पे so that is the frame shift mutation. Now we come on to the chromosomal mutation. What is chromosomal mutation? तो हम लोगों ने define किया that what is chromosomal mutation. So the change in the number or structure of chromosome is called chromosomal mutation. So ultimately here we are using the word two terminology, number or structure. So ultimately the two variations are there. So we just talk about for the first, when there is a change in the structure of chromosomes, or we can say when there is a differences or variations in the morphology of the chromosome. So that type of the variations is called as the structural variation. When there is a change in the morphology of chromosomes, chromosome की whatever the structure is there of the chromosome, उसमें changes आना. How these changes takes place? There are lots of the conditions, either duplication, deletion, inversion or translocation. These are the four main conditions by which there the structural variations are there. अब structural variation, chromosome पे जो genes हैं, these genes की conditions में variation आ जाना. Simple. For example, हम लोग बात करें that we are having a chromosome. This is the one chromosome we are having. On this chromosome we are having some segments of genes like segment A, B, C, D, E, F and G. That is the one of the chromosomes we are having. अब इस chromosome पे क्या हुआ? ये first we talk about the duplication. A, this B is repeated twice. E, F and G. तो यहाँ पे क्या हुआ? ये जो B वाला segment है, this is repeated twice. तो duplication हुआ. This is simply called as the duplication. Now we come on to the next. With the same case, if we say in terms of the deletion, the same chromosome है हमारे पास. Now in this case क्या हुआ? A, this B has been deleted. B or has been deleted. So C, D, E, F, G. This is called deletion. अब deletion in this case क्या होगा कि ये जो B वाला जो segment है आपका, this B segment is deleted over this side. So that is the deletion over here. अगर further अगर हम लोग बात करें the third condition, that is the inversion. This is the chromosome. ठीक है, अब यहाँ पर क्या हुआ, inversion inward. A is there itself, instead of B now it comes D, then C and then D. That means these are inverted. C is remain on its position, but instead the B and the D replaced by their positions. So it can be of so many types. एक चेंज भी हो सकता है, दो हो सकते हैं, बहुत सारे variations हो सकते हैं, and then E, F, G, that will be the remain same. But this is the third category. So we just talked about for the duplication, deletion, inversion. That's it. Now in terms of the translocation, we just talked about for the translocation. For the translocation, we just take two examples for the two chromosomes. Okay, so this is the one chromosome and the another chromosome contains P, Q, R, S, T, U, V. That is, अब इनके क्रोमोसोम में क्या हुआ, there is a change. For example, we are just talking about for the single chromosome. In this, along with P and Q, here the B and C are also added. Then the remains are same, R, S, T, U, V. So that means अब ये वाला जो पोर्शन था it will be half. इसमें क्या होगा, the B and C are not there. So that is translocation. That some genes are translocated from one chromosome to the another. So there are so many reasons. They may cause some of the diseases as well. They are not supported. They are not expressible as well. So there are so many conditions which can be defined as structural variation. Now next. The numerical variations, when we talk about for the numerical variation, in the numerical variation, we define that the change in the number of chromosomes. Numerical, by the word itself it is clear that it is defining the change in the number of chromosomes. The change in the number of chromosomes is further classified. ये जो numerical changes होने हैं, these are further classified. अब now, that overall we are talking about for the changes in the genome. अब जीनोम में कहाँ पर वेरिएशंस और किसके नंबर में चेंजेस आना है, तो द टू कंडीशंस आते हैं. फर्स्ट थिंग, जीनोम कंप्राइजेस ऑफ द क्रोमोसोम्स, तो द नंबर ऑफ क्रोमोसोम हैज बीन चेंज्ड. द सेकंड कंडीशन, द जीनोम कैन बी चेंज्ड. द नंबर ऑफ जीनोम्स हैज बीन डबल, ट्रिपल एंड सो ऑन कैन बी देयर. सो यहाँ पे हम लोग बात करें तो द वेरी फर्स्ट कंडीशन दैट इज द एनियोप्लॉइडी. द एनियोप्लॉइडी व्हेन वी आर गोइंग टू डिफाइन दैट इन दिस what the things has been done in aneuploidy, the change, the numerical change in the number of chromosome in genome of an organism. किसी भी organism का genome है, उस genome में whatever the number of chromosomes are there, उसके number में variation आना is called as aneuploidy. Again I am repeating, the numerical change in the number of chromosome in genomes of an organism is called aneuploidy. For example, the simple example we can just talk about for the humans itself. हमें पता है भाई all the human beings contains the 46 chromosomes. That we know very well. 46 chromosomes हैं. अब now there is a change in the number of these chromosomes. Either the one condition it can be 47, increase हो गया एक chromosome, variation है. Whereas it can be less itself. It will be the 45. कम हो गया. Yeah, number has varied. So that is the condition. Now, how these conditions can be developed? So these are the conditions because during the gametogenesis या फिर during the process of meiosis. Meiosis हमें पता है, the process of meiosis is the reductional division. तो reductional division के टाइम पर क्रोमोसोम का सेपरेशन होना था. तो during the non disjunction of the chromosomes, तो chromosomes वहाँ पर separate नहीं हो पाए and then they can go through simultaneously in one of the gamete. So during that time, these conditions may arises. So these are can be classified as a monosomy and the trisomy. What the monosomy? By the word itself, single. Trio, three. Here mono, we are using the word for the pair. दैट मीन्स कि जो पेयर था दो होने थे वहां पे अब नाउ व्हाट दे आर सिंगल ओनली, दैट मीन्स डेफिशिएंसी ऑफ वन क्रोमोसोम, or we can say one chromosome less. यहाँ पे ट्रायोसोमी तो दैट मीन्स जो पेयर होना था वहां पे ट्रिप्लिकेट की कंडीशन एक्सप्रेस होगी, तो दैट मीन्स वन क्रोमोसोम मोर or showing the triplicate conditions. So they may cause some diseases also. So the monosomy की जो conditions होती हैं they can also cause some of the diseases, whereas in the cases of the trisomy, monosomy, the most common disease is Turner syndrome. Whereas the trisomy के common examples are Down syndrome and Klinefelter syndrome. So these are the examples for this. The monosomy, that means कि यहाँ पर क्या हुआ, the one chromosome has been less. Whereas in case of the trisomy, that means on any of the chromosome pair, कोई भी पेयर हो सकता है, either first to लेकर 23 तक पेयर ही, कोई एक पेयर shows the trisomy. Whereas in case of the one of the pair shows the monosomy because of the non-disjunction during meiosis process. Whereas the another condition हमारे पास बचती है polyploidy. Here we are talking about for that there is a numerical change in the number of chromosomes in genomes. Here what the polyploidy is, by the word itself, poly, that means so many are there. So many are there. तो यहाँ पे condition होती है with regards to the genome, that the phenomenon of having twice the number of genome. हम लोग सिंपल यहाँ पे बात करते हैं, we just talk about कि किसी के पास अगर one n condition है तो we are calling them as a haploid. For the 2n diploid, but for 3n, 4n, 5n and so on, these all are included in case of polyploidy. For the 3n, the most common example is banana. Their chromosomes we are representing with the triple set. So that means they are showing the triploidy conditions. So this is a simple example. So we are just recalling our topic once more. We just recall. We have started with the mutation. So what is mutation? हम लोगों ने define किया the sudden, stable and inheritable change in DNA sequence of an organism is called mutation. Second, we talked about for the mutant. What is mutant? The organism which undergoes mutation is called mutant. Third, हम लोगों ने बात करी किसकी? Mutagens. What are mutagens? The external factors which causes mutation. It can be radiation, it can be chemicals, it can be temperature as well. And the very first time the term mutation was used by Hugo-de-vries in the year 1903. Then we come on to the types. So there are the two types of the mutations are there. One is the gene mutation, another one is chromosomal mutation. The change in the structure of gene is called gene mutation. The change in the structure or number of chromosome is called chromosomal mutation. Then we are defining that as the point mutation and the frame shifting mutation. The point, the single nitrogenous basis के variation पे जो भी आ रहा है, that is the point mutation. And here the addition or deletion of one or more nitrogenous bases is called frame shift mutation. Then chromosomal mutation on the basis of the structural variation, the numerical variation. We just studied about the duplication, deletion, inversion, translocation. Then the numerical variations are there, aneuploidy, in which we studied about the monosomy, trisomy. Then we discussed about the polyploidy. So these are the conditions. So just remember the most common example for the monosomy, that is the Turner syndrome. Because of the Turner syndrome shows the monosomy. Then here we just say sickle cell anemia. These are some common examples which on which number of times the question has been asked. This is about the mutation. I hope you like this video. So please hit like button and please subscribe my channel too and stay tuned for the upcoming videos. Thank you.